One we did with the innate immunity, and second one is the specific immunity. And the specific immunity, we did study the cells and the cells that produced antibodies. Okay, so we have innate immunity as the first line of defense, which we have already seen that one discussed elaborately. And the specific immunity, it's specific to a particular infection, specific to a particular pathogen, specific to a particular bacteria or viral infection. So that's the specific. So this specificity comes from anyone. How do we identify the specificity? Do we have any question? Any answer? Question is, how do we determine the specificity? Okay, lock and key. What is lock? What is in key?
Antigens are the key antibiotics. Antigens are the key antibodies are locked.
No, you are saying the other way around.
The other one?
Yeah, you have the antigen as a lock, and then antibody is the key, right? So the hormone receptors. So basically, the question is, you have a specific antibody, right? And you have a bacteria or virus or any other antigen. I mean, the antigen is again a type of protein, and virus membrane, they have the protein. Bacterial membrane, they have the protein. And this antibody is specific for this, and this antibody is specific for virus. This antibody is specific to a particular antigen, which is already there in our system. Okay, or any other foreign invaders. I put it like a pollen or any other invaders. So they have the protein. So this is determining, or these antibodies, they are the determinant. So now we need to know, what is this antibody? So our question is, what is an antibody? So we are going to deal the entire class on antibody. Okay, what we are going to learn in this antibody, our objective of the class. Okay, antibody. First, we want to see the structure of antibody. I put AB for antibody. Okay, and antibody molecule. You have to remember that whenever I say in the biology, the molecule, we call it as a biomolecule. Okay, meaning it is a macromolecule or macromolecule. In chemistry, you might have studied about the molecule with the collection of atoms and elements that constitute a molecule, right? That you understand that one, the chemistry. But here the composition is mainly of this molecule is made up of amino acids. Amino acids. In other words, the collection of amino acids is going to be polypeptide, or we call it as proteins. I can say antibodies are proteins. This is the basic stuff. Antibodies are proteins, and these proteins are made up of amino acids, and they have joined together amino acid and different type of amino acids are getting a polypeptide. So this protein, in other words, they are specific. How I say it's a specific? It's specific for a bacterium. A bacterium. Bacterium is singular and bacteria is plural, right? Single bacterium. A species, a particular species of bacterium, or a specific for a viral protein, or it's a specific for a pollen protein, which is an allergic protein. Okay, So if I say specific means it will not, this bacterial antibody cannot bind with the viral, it cannot bind with the pollen, no, but only this will bind with the bacterium. Okay. Now the next step is the specific nature means specific nature of this antibody, meaning these peptides should have, how do they make this peptide? They should have originated from, how do you say, like a protein synthesis? You need to have a mRNA, correct? mRNA, when you consider a peptide, how the protein is made in our system, the message, how the sequence of amino acids are arranged, so that is in a sequence of nucleotides. Otherwise, it is originated from DNA via mRNA. Okay. Otherwise, I can also call it genes or coding for these amino acids of polypeptide. Okay. Do you follow this? To summarize this, what I have started now, innate immunity is a phagocytes, that's we covered chapter 1, and chapter 2 specific immunity, and we have studied about these how specific nature of the cells and that produces antibody. Now we are going a little further into that, what is these antibodies are, how these antibodies are made up of, what are the constituents of this antibody that's on the peptides. So we say polypeptides, where the origination of polypeptides, the polypeptides again is coded by a specific mRNA, and the specific mRNA coming from the gene. So now we know the genes are responsible for coding this antibody. You understand that part? Yes or no? Any questions from Victoria, Kelly? Okay, any questions from Cinco Ranch? Okay, good. Because just I want to make sure it's a recollection again what we are going to discuss in future now. So if I say the antibody or the peptide, then what is the structure of antibody? Okay. Again, I mean the structure which I mentioned before is the amino acid sequences. Now what I'm going to discuss with you, how they formed, otherwise the morphology of antibody. Otherwise I can say how they organized the amino acids and the peptides. Okay. In other words, how it looks like. This macromolecule looks like according to the function. Okay, how it looks like, how does it look like for specific function. In our earlier class, we did study about the specific function of antibody is an adapter molecule. Correct? So the specific function of amino acids, so specific function of this antibody is going to be one side, this is bacteria, it binds with one end, right? It binds one, this is antibody and binding region, and also we did study some of the recognition site, okay, and some of the other side is going to be an activation of phagocytes. So another one is the binding region. So this is the crude way, just I'm drawing this antibody structure so that you will understand a little bit further when we go further down onto this chapter. Okay, so these are this antibody binding region, and these are the constant region. How do we determine this adapter molecule? What is the how the people they find the structure of this antibody, which I said which region is a binding. How do you determine the binding region? How do you determine the other portion, which is activation of that? Okay, so that's the part which I'm going to explain to you now. There are two regions, two types of polypeptide. Two types of polypeptide. Otherwise, I call it as two types. Is going to be chain, heavy chain, and light chain. Heavy chain molecule, light chain molecule, and they clump together to form one antibody. Okay, so antibody is composed of two main types of polypeptide. One is a heavy chain molecule, another one is the light chain molecule, and they combine together and they form antibody. How they arranged? Now we can look into the arrangement of this chain. Arrangements of this heavy chain and light chain. For example, they said in our experiments, we will see in a little bit, the experiment they proved that the antibody looks like this. Okay, the red one is the heavy chain. Okay, and the light chain will come onto this part. The blue one. Okay, so these two chains now they linked. See, this is the one chain and this is the another heavy chain, and this is one light chain, this is another light chain. So I'll put it like this. This is a light chain molecule, light chain. They are heavy chain molecule, heavy chain molecule. So this is one heavy chain, and this is the one light chain, and then one heavy chain, one light chain, and they are linked with a disulfide bridge. Okay, I'll put it like this. Here. Okay, and this one is here. So this region, this region, I put it as they linked, they joined together with a sulfide bond, SH bond, sulfhydryl bond. This is also a sulfhydryl bond, and this is also a sulfhydryl, SH group, hydryl bond. Meaning there is a sulfhydryl amino acids are present here and here and here, and they join together with the hydrogen bond on this. So in total, the antibody molecule looks like this. So if I say the antibody molecule, then you have to understand that the two heavy chains and two light chains. So how many chains are there total? 1, 2, 3, 4. There are four polypeptides. They join together, joined in a fashion with sulfhydryl group, SH group, sulfhydryl group. So three sulfhydryl group here, here, and here. They join together and held together. Okay. Do you follow up to this point? How this antibody molecule looks like, and how this antibody molecule is arranged, how this antibody molecule they combine with the chains, two light chains and two heavy chains. Okay, so we are seeing one antibody molecule. This is everything put one molecule, one antibody molecule. So this is the structure of this molecule. Yes. Hereafter, we are going to see this structure. Okay. One antibody molecule structure. If I ask you to write a short notes in your exam, what you need to do? Antibody, short notes. That's my question. What you need to answer? Antibody are made up of proteins, and they have specific gene sequence, and antibody molecule is made up of 4 chains, 2 heavy chain molecule and 2 light chain molecules, and these molecules are arranged in a way they linked with sulfhydryl bridges or sulfhydryl bonds. This is the, so far which I taught you about this antibody. This is the introduction to the antibody. Do you understand that? What is antibody? Because this is the entire class is made up of this one, and then we are going to deal further detail of what is this, what is this, what is this, what is light chain, what is heavy chain, and function of this, what kind of antibody, all those things we are going to discuss in future. So just I want you to understand what is antibody now. So do you understand that, this part? Great. I'll go on to the next one. I'm going to draw the same way so that you'll follow that. The lines is easier to understand. A larger piece, and Y shape. I just put a drawing like this. Okay, because they're linked with this one, sulfhydryl bonds. Okay. There are different types of the proteases. I put it in a way that if you digest an antibody, meaning this peptide is liable, they are more vulnerable for protease enzyme, because they are protein. So if you add a particular type of protease enzyme, it will cleave this one. So for example, to test the different region, and people, they are scientists, they have already done some experiments. Based upon that experiments, we can study individually on this one. Okay. We'll see with the first one, which I put like pepine enzyme. Pepine. This pepine is coming from papaya fruits. Have you eaten papaya? There's a lot of proteases in that one. Okay, so if you have any type of meat indigestions and everything, if you eat a lot of meat, it's advisable you eat along with the meat more of papaya fruits on that. So the papaya, on the skin of the papaya fruits, a lot of pepine enzyme, that proteolytic enzyme, that will help digestion in your system. And in this case, if you have a purified pepine and then add into the antibody molecule, it will cleave this molecule into two like this region. So there are two fragments which is coming out here. This one region of this, we call it as a constant region, we call it fragment FC. Okay, this region, FC region. And this region from here to here, it calls a fragment of antibody binding site, otherwise FAB. And the same thing is FAB. Fragment of antibody binding. These are the region, and these are the constant region. Any antibody molecule, because we are going to see different types of antibody molecule. And this region, which is a constant region, as you recollect from the last class, we discussed about this constant region of this adapter molecule should have a number one communication step, and another one is the activation of phagocytes. Communications, otherwise complements, inducing the complements. So there are two functions of the constant region. Any antibody will do the same function. So this structure, we call it as a constant region of antibody. And this region is like a clamp-like thing, right? You have a clamp, just you can hold it like that because of the two region over there, this molecule. This molecule is antibody binding region. So this is the one with the binding region is with the bacterial or antigen, bacterial antigen or viral or pollen. So whatever you have, so this region will go and bind on that binding region. So this region is not as a constant region, it is a variable region. I put it over there. FAB is a variable region, variable region. So now I will summarize of this slide what we learned so far. Antibody molecules are made up of 4 chains, 2 heavy chain and 2 light chains, and they are linked with sulfidal bridges. When you treat with the papain enzyme, is a proteolytic enzyme, it cleaves into 2 parts. One is the constant region, the other region is the antibody, F for antibody, FAB. So if you have 2 FAB, so FAB2 molecules here and 1 molecule of constant region. The function of the constant region of any antibody, this sequence and this region is same. It's a constant because it will activate phagocytes, also it induces the complement. And these 2 regions of the FAB fragments are the variable region, which will bind to any of those antigen derived from a bacteria or viral or pollen or any other antigenic sites. So this region is a variable region. It may vary bacteria to bacteria, viral to viral, or any other specificity. So this region is more of the specific binding region. So any antibody molecule, when you look into the genome level or genetic level, this region of the sequence of the DNA will be the same, only this region will vary, and they form in a very good fashion of the linking of this peptide to form one antibody molecule. And when I say that, do you understand that up to this point? Can I go a little bit further? It's okay? Syncorange? Victoria? Do you follow now up to this point? No, I couldn't hear you. Syncorange? Yep.
Yes, sir, we follow you.
Oh, okay. All right. So now I want to go in a little bit further onto the genes. We will study more on the genes on that, but to go just to say a constant region and genes for variable region. So when you see that these genes are different, but when they combine together, they form antibody, one antibody molecule. Okay. Sequence, the gene sequence for the constant region and gene sequence for the variable region. Okay. They form one antibody molecule. Yes. Now, I'll ask one question now. Variable region, how many chains are there in variable region? Yes? 4 chains, good. So 4 of what? 2 heavy and 2 light chains. That's good. So these 2 heavy and 2 lights, you can say identical twins like, isn't it? Identical, 2 identical heavy chains and 2 identical light chains. So these heavy chains and light chains, they have got the same region onto the variable region here. And then you have one pair of the constant region. Okay? So if you combine this constant variable region, then you form an antibody molecule. Okay? So the combination of this variable region, how these genes are being taught, hey, there is a new virus or new bacteria and this is the polypeptide. How do they know I have to synthesize this particular variable region to bind? How do they know what language they speak, when they will learn? So that is the core of this immunology part of this your class. I mean, that's the part which is the crosstalk between a pathogen and your immune system, and then what cells are involved, that we will see a little bit later. But now I want to summarize this. I want to go into the PowerPoints, and we will look into the little bit details and different types of antibodies and what is the functions of those antibodies, where those antibodies are present. We are not talking about only one type of antibody. As I mentioned, there are some antibodies are responsible for our innate immune system, and some of them are a specific immune system, and some of them they acting both ways of activating the complements and helping this innate immune system. So all of them, packed together of this antibody chapter. Okay, I'll go onto the PowerPoint, and then we will go into the details. Okay. Could you see that one now? Here in SynchroRange? Slides? Yes? Okay.
Yes, sir.
Okay, great. Kelly, could you see in your Victoria projection is okay? Is clear now? Okay, good. So the immunoglobulin, we call it as an antibody. In other words, we call it in our system, we call it IG, immunoglobulin. Okay. Have you done in your blood chemistry of albumin, blood composition, albumin and globulin? Yesterday? No, you haven't done it yet. So there are some ratios on that albumin ratio, the protein, and there are different types of protein in that in our system. So AG ratio, albumin, A for albumin and G for globulins. That ratio determines the thickness. That ratio determines the how much protein you have. You have a total protein. Some of the proteinuria, people they lose more protein when there is a kidney infections or kidney damage. So the protein present in the urine. So normally the doctor's office, when they were the urine, void urine, and then you check the protein measurements or protein level, it should not be present any except the Benz-Jones protein and some other specific protein with a very low molecular weight protein is excreted in some certain type of diseases. But otherwise, protein is not to be present in the system. If it is there, then there is something wrong in a urinary pathway, as well as any damage in on the kidney or urethra or urinary tract infection or any other prostate or anything. So you may get some other protein present in the urine. So that's the way they lose more of the protein in the blood, and thereby they will check the globulin level as well. So what in our system, in our class, you should understand that antibody, in other words, in another name, we call it as immunoglobulin. The protein which will protect us like an immune system, you should remember like immuno, our protection, and globulin, a protein. Which protein will protect in your body? Antibody. Do you understand that? So you should not get confused on that one. Immune, okay, is a protection. Globulin is a protein, the protein which protects. So that's why immunoglobulin is the one is something like antibody now. Okay, so structure of the molecule is two identical heavy and two identical light chain. This I'm summarizing this. Held together interchain disulfide bond, abandoned in type of antibody is a IgG. This is the one which you should understand then. As I mentioned before, there are several types of antibody in our system. Okay, so what type is majority in our blood, in our system, is IgG. Okay, immunoglobulin G, we call it immunoglobulin. I stand for immuno, G, small g stand for globulin, and G is a type. IgG, we are going to see a different type, IgG, IgA, and different types of antibodies. So we will say that immunoglobulin, I put immunoglobulin G molecule, is predominant in our system. Presence of high vulnerable proteolytic attack, which I mentioned before. These are the immunoglobulin molecules that are attacked by the structures of papain digest to give two identical Fab fragments, and each Fab fragment with a single combined site for the antigen site. See, site for antigen means the antigen binding sites. That's the one which I just mentioned before. Just I want to give you a clear. So this region, antibody, here the single binding site, and this one single binding site of this antigen. Okay, antigen binding site. Third fragment, FC, that lack the binding to antigen. So this spot, this region is not binding to antibody. Okay, so that you should understand clearly. Whenever you say the antibody binding to bacteria or viral or antigen, I am referring to this region of antibody, Fab region, where it have a binding capacity. And here it is not. This is a constant region. It is not going to bind with any antigen. This is antigen not binding. FC region is not binding with antigen. That you should understand. Okay, the pepsin attack, which I just mentioned before, two Fab fragments and 5s fragment, that's called it, and that the molecular weight, and different point of attack cleave from the remainder of the molecule FAB2, the divalent, just like the parent molecule. Here the divalent molecule is something like just mentioned before, it will bind these two to these two region, which is hypervariable region, and this variable region can bind to the molecule. It's a divalent, and here also is a binding. So two type of binding. That's a divalent molecule, just like a parent molecule. Then the amino acid sequence, that reveal the variations. So why there is a light chain and a heavy chain? Why I say light chain and heavy? Where the difference is coming from? Coming from different in variations. The light chain is having a type of amino acid, a series of amino acids, is different from the heavy chain. That constitute the heavy chain and light chain. Okay, so that's another region. And immunoglobulin structure, we will see that one. And there are 10 to the power of 8 or more different Ig molecule, immunoglobulin molecules in normal serum. How many? 10 to the power of 8. Yes? Yeah, there's a lot. There's a lot of immunoglobulin. Okay, different types of this immunoglobulin are present. That's why we have a protection. See, now we know, according to the evolution, okay, how the man, the age is going to be millions of years or 4,000 years, including if you get the organisms, say, okay, they say millions of years, okay, according to the the latest history, say, okay, 4,000 or 5,000 years, or thousands and thousands of years, we can say that. But still, the human race is going on. Do you think those time which we never ever have the knowledge of defense mechanism or immune system or vaccination, still people survive? Do you understand that? That's why we are here. If human race is gone with the big infections going on, then we may not be here, right? So those molecules, 10 to the power of 8, that thousands of molecules, so that's the one which put through in us to survival for the different struggle with the any other pathogen through the human history, human race so far. So now we know the science and knowledge, so that we are going back and then find out the mechanism. So now we elaborate, we know what is what, and we can prevent and protect us in a scientific way. That's fine. Even if you don't treat or anything, yes, people, if they want to live, yes, they are living for a long time. And it's not being dead and gone. Major impact is this molecule, 10 to the power of 8 more different Ig molecule in normal serum. In normal serum, you have this already existing antibody in our system. Okay, so that's one information just I want to share with you. Then we'll go on the next one. If you, one example here, the myeloma proteins. Myeloma is a type of cancer. So when you look into that particular protein molecule, homogeneous is only one type of immunoglobulin molecule in the myeloma protein. Though there are other types which I mentioned, 10 to the power of 8, in among them, this is the majority of the protein which is there. Why? Because it's the single clones of malignant plasma cells. The malignant plasma cells that will produce more of a one type of protein, that's myeloma proteins. The one type of Ig, that's immunoglobulin. And that's a variable amino acid sequence found in N-terminal region of heavy and light chains. So this N-terminal, how many of you have studied about the protein structure, N-terminal, C-terminal? Most of them, you know about it, protein structure? Or I can go further and then explain to you, and then we will come back to this one. 1 second, I will recollect those who have understood already, but I'm just maybe a revision for you. I'm just giving an outline of the protein structure because that's very interesting one, okay? If your proteins are made up of amino acids, correct? Amino acids. And amino acid 1, and amino acid 2, and amino acid 3, and amino acid 4. So you have a different amino acid, they form a linkage, okay? So the amino acid structure is made up of, I put it like that, and NH2, CO, and then R, and then H. Like this is one amino acid. They put R alkyl group or any one particular RO. So another one here, this is the polypeptide linkage, CONH here, this bond, and then another molecule, R2, and then H, and then CO, and now NH, so here the NH molecule, NH molecule. This is from the, this NH is coming from this amino acid. This is for the one amino acid, this is another amino acid, this is another amino acid, NHC, R3, CO, and it is going on. So this is going on and on. So this is amino acid 1, 2, 3. So when you see the structure of amino acid, this is the one, the nitrogen group, right? This is the free nitrogen group. And this we call it as a N for the nitrogen or amino group, N-terminal. And when you go add more and more amino acids, and the final amino acid, they will have a CO group at the end, or the free one, that free CO group, the carbonyl group. We call it as C-terminal amino acid. For example, this is a 1 and this is a 300 amino acid to form one peptide. So first one is the that's an N-terminal, nitrogen terminal. Another one is the carboxy terminal or C-terminal. So whenever we address a protein or a polypeptide, we normally address the amino acid which is closest to the N-terminal, which is closest to 299, which is closest to C-terminal. Or if you say in the middle of 150 amino acid. So whenever you want to identify a particular place, what you normally do? New York. Is New York is closer to Washington DC or New York is closer to Houston? Which one? Huh? Washington DC, right? New York. So how do you know that? By the map, and then you see that, right? The same thing is here. Whenever if you want to identify a particular place, you need to have two variables, two regions. Then you can pinpoint where. If you see, I think the calculus or algebra, when you have two terminals for your 2 points, and then you have to identify the in-between steps, and which is a closer region, yes, you can easily identify that. You need to have 2 coordinates to find out one other things. I'm not going to teach that math. Okay. Now you understand this molecule with either the C terminal and then the N terminal. That's what my point is. So the molecule, if you say the protein molecule or polypeptides, it has one terminal and this is another terminal, so that the entire molecule can be identified which amino acid closer to which terminal or not, to identify for our mutations and other part of the thing. Any questions on this? This is a basic protein chemistry. Okay, probably you might have studied with a biochemistry class, right, with Dr. G, right? So it's good. If you don't understand, please ask me, and then I will go into that detail. So in the PowerPoints, variable amino acid sequences found in N-terminal region of heavy and light chains. Okay, now coming to this point here, variable amino acid sequences. Okay, now I put like a heavy chain and light chain. I put like this, right? Heavy chain, and another one is the light chain, right? Light chain's coming out. And this one, variable sequences, variable amino acid sequences, right? They are present in both region, terminal, here, heavy chain, and then here light chain. Where it is an N terminal. Suppose if you have an N terminal, this is the N terminal, this is the one. The N terminal. The C terminal is not that much over here. The C terminal of this is C terminal. So the variable region has only the tip of that one. And then the constant region is coming out. This one is a C terminal, and this C terminal is going to be attached to the other constant region. So we don't want that one. So here, this is the region. N terminal region is that one. That's what just my point. So the variable region of the amino acids is here, and that's all the amino acids. Okay. The remainder region is constant in structure. This part is a constant structure towards C for the carboxy terminal and C for the constant region too. Okay. Now we go on to the immunoglobulin genes. Okay. Which I mentioned before. These genes, the proteins of antibody peptide or polypeptide, they coded by the genes. Now we will go into certain type of arrangement. The organization of this polypeptide. Do you follow that one? Organization of the polypeptide. Clusters of genes on different chromosomes. Encode, you have alpha and beta as well as the AV and the chains, heavy IG chains. That's a heavy region. There are different type of different region in the immunoglobulin chain, the immunoglobulin molecule. I think you don't get on that one. Probably you can check an alpha and gamma region, which is not coming here in the PowerPoint. 30 to 50 functional variable region, we call it V genes. Functional variable gene, V gene, present in each cluster. What is the cluster now? I'll just explain to you in a little bit. Go on to that. So you know about our cell, nucleus, and you have chromatin, chromosomes there, right? There are 46 chromosomes or 23 pairs of chromosomes over there. So if I do on that, 1, 2, 3 like that, it goes on. Okay, I'm just spreading a karyotype of these chromosomes. Okay. So the variable regions, and we are talking about different types of variable region or the binding region of the antibody, and they coded with the different type of genes, and these genes are coming from the cluster of genes from different, see, one cluster here, another cluster here, another here, and another here, and another here, another here. So you get different part of the chromosomes, different chromosomes, they have the cluster. Okay, and these clusters, they code for IG chains or immunoglobulin chains. So I mentioned before, the immunoglobulin chains, they made up of amino acids, And these amino acids are then organized from the genes, and the genes are not from one chromosome, and they come from different chromosomes. They're coming from different chromosomes. "30 to 50 functional variable regions I just mentioned before in each cluster." If I say one cluster, okay, cluster 1. So the cluster 1, for example, it may have, this is a chromosome 1, 2, 3, 4, 5, 6, 7, and 20, 21, like that, okay. The cluster 1 will have probably chromosome 2, chromosome 5, and chromosome 7. Cluster 2, they may have 3 and 5 and then 9. And then cluster 3 of the variable region of, you may have 5, 9, and 7. So if you see the combination of each cluster, these coming from different chromosomes, and they form this cluster. Just giving an example for you to understand simply, because the ways we will go into that a little bit later, but just I want to make sure this is the cell, and you have the nucleus. Inside the nucleus you have chromosomes. If you spread out the chromosomes, and each chromosome will have their own cluster unit, meaning they code for antibody protein of the chains. So each chain of one chain of antibody, in other words, I put one chain, I put the one chain, okay. This is the one chain of, for example, is heavy chain, okay. Heavy chain. It will have a cluster, different cluster. So this amino acid sequences is coming from cluster 1, cluster 2, and cluster 3. The another region of heavy chain, of variable region, may be coming from cluster 1 and cluster 3 only, not 2. Another one will come from cluster 6 or cluster 5 and cluster 1. So one chain, but this one chain of the heavy chain or light chain, or one chain of immunoglobulin, and they have the amino acid sequences derived from different clusters. And each cluster is made up of a different region of chromosomes. This is something like a permutation combinations of those things. So each chromosomes we have a different genes on that one, right? So it's code for different type of proteins. So here in this case, we have to remember that pick up from 1. Don't think that chromosome 11 is coding for a particular antibody. No, the answer is absolutely no. Though there's some region, some genes coming from chromosome 11, some of them coming from chromosome 2, some of them coming from chromosome 3. So chromosome 2, 3, and 11, and put together one cluster, and that form one particular heavy chain. See, another region which is going to be chromosome 6 and chromosome 9, chromosome 21. So from that genes, they code for the protein, and that protein form another heavy chain. So each heavy chain will made up of a different genes from different chromosomes. That's the point which you'd understand. Do you follow?
Dr. Soma,
Yes, go ahead.
the circle below AA, what is in the circle?
Which one? This circle? This I put a cluster 1, 2, 3.
The red one.
Oh, the red one is the cluster. The genes which is coming from here.
Lower.
Oh, here. Oh, here. This is amino acid. The genes code for amino acids. See, just I put,
G.
yeah, this is a gene. This is a gene. And these genes code for the amino acids, right? So these genes are the different chromosomes. Genes,
Okay, thank you.
yeah. Different chromosomes, the genes from different chromosomes constitute for one antibody molecule. Okay, I put it in a simplified way, in other directions on that. Suppose if you have a albumin protein, albumin. Okay, this is the one protein. Okay, one single protein. So you have its own chain molecule. These are the N-terminal and C-terminal. You have amino acid sequence, and this is called albumin. And this albumin is coded by a chromosome, for example, chromosome 12, and that region is there. So this particular region of the chromosome 12 will code for albumin. Period. Over. Chapter close. One protein, one gene, one protein, over. That's a strike. That's what we learn from central dogma of molecular biology. DNA, RNA, protein. So one DNA from here, and this one mRNA for the albumin mRNA, and then got one protein, that albumin. That we know everybody will understand that point? Yes? Right? You get it or not? One gene, one RNA, one protein. That's fine. But here, in other words, in this antibody molecule, if you have this one antibody molecule here, AD for antibody, okay? This particular region, one gene, gene 1, gene X, I put it over there. And then here, this one, gene Y, and then this region. And this region is coded by gene Z. And this gene Z is coming from chromosome 11, and this is chromosome 5, and this is chromosome 1. So, in a nutshell, I can say that this is coming from gene 1, gene 11, and this particular region of the protein which is coming from here, the chromosome 5, and this one is coming from gene Z, and that is coming from chromosome 1. So when you see these chromosome 11, 5, 1, they form a gene, and this gene, as we call it, as a cluster, and this cluster will made on these particular amino acids, and that constitute an antibody. That's a one protein. This is one protein molecule, antibody protein molecule, one protein. So antibody protein is different from a regular protein. That's the message you should get it. There are more genes are involved for the formation of one particular polypeptide, antibody polypeptide. Not just like one, like albumin or just one for one. No, it is not one for one. Here, too many for one. I put it like that, right? I put not two, I put many genes, one protein. That protein called antibody. Okay, many genes, one protein, and one antibody. Is that a message? Yes? Okay. Just meditate on that. I'll give you a break for 10 minutes, come back, and we will go further. Okay? Any question? No question? Okay. One antibody specifically binding to one specific bacteria or virus. But this antibody is derived from not one genes, many genes, and that is derived from many chromosomes altogether. Okay, so that's the part you have to understand in this. And then what I'm going to do, I'm going to go on the different types of chains and the segments, and we call it as a functional variable region V genes present in each cluster. So that's what it constitutes, which I explained to you. They also have 4 to 6 small J mini segments. Okay, and the immunoglobulin, they have heavy chain clusters in addition to V. These are V and J heavy chain clusters in addition to that. And also they also contain 25 D mini genes. So they also code. So how many genes are there here? B genes, J genes, and D genes, right? And they also have a constant region, as I mentioned before. In the immunoglobulin structure, you have both region with the constant region as well as the variable region. The constant regions, and they can also go for a single gene encoding different class and subclass. So we will see about these gene class and subclass. So this will determine the type of immunoglobulins. As I mentioned before, the immunoglobulins are not one type, there are several types. How these types variant, that comes from these different subclass and class of those things. And functionally, these genes or these peptides or this protein or this antibody is different from each other. It's not the same function. No, it may vary. Each antibody is having its own function. Let us see on the variable or variety of immunoglobulins or Ig types. Okay, so they form. How do they form from one chromosomes and one gene or one polypeptide? But the combination vary to form a different type of immunoglobulins. So as I mentioned before, many genes, they form one antibody, and the combination may vary. So if you have the antibody X, they may have gene 1 and from chromosome 11, for example. Then antibody 2 will have the same gene, but it also have some other gene from another chromosome, so the function may vary. So that's the only point. But how do they recognize this, or how do we study? We have to study with the splicing mechanism. Special splicing mechanism involving mutual recognition of 5' and 3' flanking sequence. So the 5', 3' flanking sequence are from DNA sequence. Okay, 5' region. Now I have to go a little bit of molecular biology of DNA. How many of you study of the structure of DNA? 5' and 3' region. All of you know about it, yeah? Where the DNA sequence is coming out. Catalyzed by the recombinase enzymes effects of DJ, VD, VJ, and translocations. Please refer to your web link and somatic recombination animation. I will show you in just a second. And before that, just I want to make sure and how that many genes form one polypeptide. Just I'll give you an explanation for this. These many genes on one protein, how this many genes is organized. So that's what the question today. Now, so I just mentioned before this DNA structure. We have a 5' region and 3'. Okay, so this is the structure of DNA. If you have DNA, you have this one is going on, and you have a double helix and DNA double helix, and this is the one strand, is another strand, and this one is the 5' region to 3' core, and this is the 5' to 3'. This is the anti-parallel structure, 5' and 3'. So there are certain type of the recombinase enzymes will split here, and that will give you a strand on that. And this one again with the recombinase enzyme, which will this is from, for example, this segment I call it as a D segment. Okay, another segment which is I can get it from a double-stranded DNA. Another one we got the J segment. And this will also get, this region will combine, and this is coming from J. Another one, I just coming from a V region, another DNA double strand, and then a cut, and then it's coming out. So V region over here. So what you have, one for the J, another one for the D, another one for V, V, D, J, segments. So when you combine this one as a recombinase enzyme will link all this together to form one single DNA sequence. And when this one will combine with the RNA polymerase enzyme, which will code for mRNA. And this mRNA again on the translation, and that will give to a peptide protein. a protein. So this region, J region and D region and then V region, they contribute a fragment of small DNA, and that everything put together and to form a polypeptide chain, where all exist in one single cell. Why? Because all these are regions coming from different chromosomes of the same cell. So one cell is responsible for that on this, the whole synthesis of antibody. Do you understand that part? Okay. So now, if you go back onto your PowerPoint, the catalyzed by recombinase enzymes effect, you have a DJ, VD, VJ translocation. Okay. So when you think this combination of this, I'll just put it over there, like VD, this is one, another one is a DJ, another one, and then VJ. So if you had a combination of these things in a different fashion, so this is all three, V, D, J. In some of them, you may join only on V and D can join together. In some of them, you have a J and D can join together. Another one is the J, D, and then JV. You can comment. So you have the same chromosome or the same gene, but the combination of the gene varies, and thereby it will give what type of the antibody. If I mention just a few slides back, we saw that 10 to the power of 8, do you remember that one? 10 to the power of 8 immunoglobulin molecule or different types of antibody molecules are there. We do not have 10 to the power of 8 genes in our system. Just I want to make sure. We don't have these genes. We have only 20 to 25,000 genes. That's all we have it in our system. But how come the 10 to the power of 8? So that's why the people, they thought initially one gene, one protein, one gene, one RNA, one protein. So they isolated the protein. Hey, we have got 100,000 or 200,000 proteins. We might have 200,000 genes on that. That's what they thought initially in the science thing. But in 1999 and then 2000, they didn't find that many of genes there. So they found only very limited. Then the next part of the research is how do they come? Then they found out the recombinase and all these things, that the different combinations of this splicing mechanism. So that's what we call it as the splicing. So we call it as this DNA sequence, this splice and this splice and this splice. So they splice in a different regions. Even the RNA of this splice, and then they join together, or DNA splice, and they come recombinate, and they form a new type of protein. And this form combination, imagine how many combination you can make with the different genes and different chromosomes, they combine together. So that is the result, and that is one good example is antibody formation with the different genes and different chromosomes. So this is no, in nature it does not exist 10 to the power of 8 genes, no. But you have 10 to the power of 8 immunoglobulin. The reason is splicing, the mechanism of splicing, how they splice the DNA, different region of DNA, different type chromosomes, and they combine together to form a new protein. Yes? Okay. Now we'll go on to the next one. Structural variants of the immunoglobulin molecule. Okay. They call it as a different types. We call it isotypes. We call it variants. You know, isotope. Have you ever heard of isotopes? Yes. Same molecule, but they have different molecular weight. The same number, atomic number is the same, but that molecular mass, it may vary. Correct? Is that right? So that's isotope. So when they release certain electrons as a radiation, then they come back to the another mass. So that's why the same molecule, but it differ. It's the molecular weight. That's called the isotype. Now we got here the same thing, isotope. That one. Here the isotypes the same function of that, but they got the different variants of that one, isotypes. We will see that one based on different heavy chain constant structures, and also immunoglobulin variants are present on each individual immunoglobulin structure. So this Ig variants, it's present in a way that they have a IgG type, IgA type, IgM type, IgD type, and an IgE type. So IgG, IgA, IgM are immunoglobulin G, immunoglobulin A, immunoglobulin M, and immunoglobulin D, and immunoglobulin E. In the diseased state or any infection, this level will vary. Among them, we study IgG is the most abundant in our system, because IgG can migrate from the blood cell or capillary to extracellular space on that. So it may go from one place to the other easily, and then it can, all the synthesis or the abundant structure or the more you get IgG level in our system is more. We will see that where do you find the IgG in our defense mechanism in our system as an innate immunity. Let us see one by one. Allotypes are heavy chain variants and encoded by allelic alternative genes at single loci. The loci of what is called the gene locus, where the gene is present in the chromosome, the single loci. They are genetically distributed in the IgM group. Okay. So this is the allotype of variants. The allotypes are heavy chain variants. That you should understand. Allotypes are heavy chain variants. Okay. In your textbook, if you see on one of this one table, I couldn't see that one here. So you have the table 3.1. I think this is the 10th edition book. If you see or in your textbook, you find on the different isotype variants. I'll just focus it and zoom in. Okay. Here, as I mentioned before, this variable region, heavy chain and light chain, VH and L, this is the light blue color, and this is a constant region of this antibody. And they have the isotype here. This isotype, what type of isotype they have? They have allotype. This is present only in the constant region. And another one on the variable region, you have this isotype, we call it as a idiotype or hypervariable, or the combination site over here, idiotype. Okay, there are two isotypical variants. There's one is in the constant region, isotype is allotype, and then another one is the variable region isotype, we call it idiotype. So you should understand isotype, allotype, and idiotype. Okay, the idiotype is the one, the variation sites or the combination site, and that's the hypervariable region you get. So these are the structure, how organized they are. Okay, allotype, they are mainly present in constant region. If I ask you a question, allotypes are present in a constant region or in a variable region, then you have to say it's a constant, present in constant region. That's an answer. Okay, idiotype is specific for variable region. It is not present in a constant region of these antibody molecules. Okay. Did you get it? All right. Now, let's go back to the document camera and just go on mine. Okay, here. It's coming back here, this original picture. Yes. Go ahead.
What did you say that table was?
The table is 10th edition 3.1, summary of immunoglobulin variants. That's what I want to.
Okay, thank you.
Okay, great. So now, this is the region which I want to mention before. Just I want to recollect again the same thing what I told you earlier. This heavy chain. This is a heavy chain, right? Heavy chains, and this is also a heavy chain, right? And this is the variable region of the heavy chain, right? So this variable region of the heavy chain and this is the constant region of the heavy chain. The constant region of the heavy chain, they have what is called, we call it the isotype. What type of isotype they have? This isotype we call it as allotype. Allotype, allotype, allotype. I'll just focus it again. Okay, here, allotype. Only this part. But you have here this region, Fab region, variable region, both heavy chain and light chain, heavy chain and light chain, heavy chain and light chain. Both, they have a group called, which is the variation, we call it as idiotype. Idiotype. This idiotype is the one where this N-terminal region, where you have the combination. This is the one responsible for the binding to the bacterial antigen or viral antigen or any other antigen. So this region are mostly responsible for binding. And this one is the constant region. So allotype is present in the constant region and idiotype is present in variable region. So that's a message which I want to remember now. Okay, the next one. Idiotype. Idiotype is a typo. Idiotype is the collection of antigenic determinant of an antibody. So antigenic determinants of an antibody is the idiotype. So now I am cutting back to the original, but only thing is the terminology. Okay, as I mentioned, as a innate immune system, specific immune system, you have got antibody, and the antibody is made up of heavy chain and light chain. Now I'm going to narrow down to the hypervariable region, and the hypervariable region specifically to idiotype. Do you get it? So idiotype is the one, is the variable region. Idiotype is the one who will sense that the part which sense, or that's the part which will bind to the antigenic determinant. So that part is very, very important, idiotype. Okay. Then associated with the hypervariable regions. So why is a hypervariable region? So this region may vary depending upon the bacteria, depending upon the virus, depending upon a different type of the antigen or the any other pollen or any other antigen present in our system. So this is the variable region. And they can recognize this variable by antigen specific receptors. Okay. Now I have to recollect in a different way. Okay. Now you know, I assume that you understand like a antibody, and also you understand the antigen. The antigen is coming from where? Bacteria, or the antigen is coming from virus, or is coming from any other pollen or any other pathogen which is enter into our system. This is the antigen. This antibody which will bind together to the antigen. So that's why we want to attack this particular pathogen, and thereby we can destroy through phagocytosis. Okay. Now this antibody, now we are discussing today the antibody with a hypervariable region, region. And these hypervariable regions, which is present in the antibody on the which chain we have it? Variable region light chain and variable region heavy chain. They have it. So otherwise we call it, this region we have called it idiotype. So this idiotype can bind either with the antigen from bacteria. Why? How? Because they have the complement pair. Because we did study before receptors and hormone. So there is something like a lock and key mechanism. So they combine together, there's a complementarity, this specific antigen and to the specific idiotype. So this is the complementary pair. Okay, so like a DNA, A is combined with the T, and the G is only combined with the C. This is like a mathematics. 2 times 2 is 4. 4 times 4 is equal to 16. Like that, the combination of these is something like exact because of the complementarity of the A and T, G and C. The same way, certain type of amino acids are present in amino acid 1, amino acid 2, like that, amino acid 3. So you have the amino acid variable region of this amino acid is complement to the amino acid Z, amino acid X, amino acid Y of the antigen, this is coming from bacteria. So this is a combination. So then you have a tight binding on the complementarity. Now I want to go in a little bit further. It is not only binding with this antigenic site of antigen only, but this antibody, these amino acids, or this protein, I put like a antibody protein, they can also bind with the receptors. And these receptors, if these receptors is having like an antigenic determining sites, like just like an antigen, just like your bacteria, you have got this receptors, this antibody can easily bind to these receptors as well. But these receptors are present in our system. The receptors is not a pathogen, and it is not a bacteria, and this receptors is not derived from viral, but it is existing in our own cell, some type of cells in our own system. They also have this antigenic determining region. So they can also bind with this idiotype to the bond. Do you understand that? Now I'm going just to go one step further, right? Before I said antibody can bind only with the antigen from bacteria or virus or pollen. That's fine. Now I'm saying antibody, the idiotype, hypervariable region, can also bind a receptors of the cell in our system. There are certain type of receptors already there. So that's what just I want to remember that. So this antibody can recognize by other antigenic specific receptors. Okay, so they are either antibody, the anti-idiotype, or T cell receptors. We are going to see much on the recognition of antigen on the T cell receptors a bit later, but now you understand the cell which I mentioned before in the document camera which I showed here. They call T cell receptors, thymus derived cell, and that receptors can also bind with this antibody. Okay, now this all, if I ask you to write a short notes, I want you to write all these 4 points of idiotypes, what type of idiotypes and where you can determine. So idiotype, they have receptor, they can also bind with the T cell receptors, idiotype. And the idiotype can also bind with the bacterial antigen. That's also, it can bind. And the idiotypes are hypervariable region. They also determinant of variety, determinant of variation of the antibody. Okay, so that's the idiotype. And then the structural variants, that's the next part, is the variable region domains. They bind with the antigen. The 3 hypervariable loops, that's on complementarity determining regions on heavy chain, right, and 3 loops of light chain from antigen binding site. Okay, now we have to go on to the structure of the antibody. Okay, I'll just explain in a little bit. I'm sorry, just I want to summarize. This is the one I want to explain now. Okay, structural variants, the variable region domain bind antigen. These variable region domains which bind with antigen, where this antigen is coming from bacteria or virus, and then how they bind, they have 3 hypervariable loops. Because of the protein, they have different type of region. We call it as a loops, and that is the complementarity determining region. Otherwise, we call it as CDR on the heavy chain, and 3 loops light chain form this antigen binding site. So how we are going to do that? Now I'm just going to explain the same PowerPoint in a very different way, so you will understand that. Okay, now as I mentioned before, as a light chain, and you have a heavy chain. Okay, and they also bind with this here, right, with the region now here, right. Okay, now the thing is, as I mentioned before, there are 3 hypervariable loops in heavy chain. Here the loops 1, loops 2, loops 3, heavy chain. 1, 2, 3. Okay, the same thing, the other loops are there in the light chain 1, 2, 3. 1, 2, 3. So these loops, I mean, this is a general structure of antibody, but these loops are the region where it will responsible for binding. How do they bind with the protein? Because of the complement. Okay, complementarity, complement E. Complement here also is the complement. Complementarity determining region. So these regions, these loops, we call it as CDR. So this is the one which present in the hypervariable region are determined. Not all the proteins not binding. There are certain loops which we have a determinant of this. Complementarity determining regions. You follow that? CDRs? Just I'm explaining to you which part of that hypervariable region are responsible for that. So I'm going a little bit for CDR. As I mentioned before, the structure of antibody and heavy chain, light chain, and the light chain and heavy chain both responsible for the complementarity binding, and where you have the complementarity determining sites are binding with the bacteria, with the virus. So they are the region, 3 loops. Okay, now let's go. Constant regions are domains of heavy chain. FC carry out a secondary biological function after binding with the antigen, and the complement fixation and macrophage binding. So this region, now go back here. So how about this is over. Now this region, this we call it as a constant region, and they are responsible for biological function, otherwise we call the FC region. There is a fragment constant, F, capital F and small c, like capital F, small c. Okay. This region of antibody binding is F, A, small a, B. Yeah, fragment A, B, right? So this region is a constant region of only heavy chain. In a constant region, do you find any light chain? If you say yes, that is wrong answer. Constant region, you have only heavy chain. So these heavy chain molecules were in the constant region, that is responsible for the biological function as a binding. After binding, this is not binding, this is binding. After binding, this region, this heavy chain region will activate complement, fixation, and also macrophage binding. I put it like a M-phage, M and then stroke like this, and the macrophage, and this macrophage binding is binding to macrophage. So this region is responsible for the activation and for the phagocytosis, another word, phagocytosis, right, on the macrophage activation. So this is the constant region. So one region is binding with the activate the complement and activate the phagocyte, another region is binding to the bacterial surface. And thereby, what is the function of the specific immunity and the antibody specific function? I mentioned before, identify and kill. Identify the thief, identify the criminal, and then you can kill it. So here the identification, okay, that's been determined by the complementarity. Here it's determined region CDR, and then once it binds, and then immediately the another portion will activate the complement, another protein activate the macrophage and phagocyte, and once that activate, it come and kill that particular bacteria, viral. Okay, do you follow that part? Just I'll give you, yes.
We're missing the bottom.
Oh, I'm sorry, phagocytosis here. So this is a constant region that will activate the complement system, activate the macrophage, and activate to kill the cells, kill the bacteria through phagocytosis. And so that's the part is the identification and killing, identify and kill. So that's a part of the heavy chain constant region. So you should write the last class which I mentioned before, antibodies are responsible for identification and killing. That's the general. That's fine. But now I'm going a little bit further. Which region of antibody is responsible for identification and which region of antibody is responsible for activation of phagocytosis? That's all. It's everything you know already. You know antibody is responsible for binding and killing. Now we are going to say which region of antibody is binding to a bacteria and which region is activating the complement, and thereby it activates the phagocytosis process. So both are the same one, but one I give further explanation on that part. Okay. So the binding region here is the both heavy chain and light chain. They have the 3 loops, and they are called complementarity determining regions, CDR. So whenever I ask CDR1, there are different types of CDR. You got CDR1, CDR2, and these CDRs are coded by, as I mentioned before, like a D, J, or V region. They are derived from different genes and different chromosomes, the different clusters. So they form this region. Okay. Unlike this may go not as a variable. This is a one type of a gene or protein which is coded for this because the constant region. But only this different type of bacteria, different type of virus, and different type of antigen, we are changing this group. It is a variable. That's why we call it not one variable or two variable. We call it hyper variable. This is all is a dynamic. It is all in a way it changes depending upon your exposure to disease vaccine, disease pathogen, and different type of vaccines. This is the one which is responsible for that. And we call it in a way, what type of isotype is it? It is an idiotype. And then we call it that idiotype, we have a different region. That's our CDR1, CDR2, CDR3, CDR4, a complementary determined site, or different region. So that's what it's all about now. You follow now? Now we'll go on to the next one. Immunoglobulin classes and subclasses on this. Okay, 5 major types of heavy chains, giving 5 classes of antigen. Immunoglobulin. So 5 classes that depending upon the 5 major types of heavy chains. So if you remember earlier we discussed about it. Okay, immunoglobulin are like immunoglobulins. I call it simply IG. Okay, this is nothing but antibody. Antibody, otherwise called immunoglobulin, and then I shorten form IG. And this is made up of heavy chain and the light chain. And this heavy chain is the one H is responsible for what? 5 types. H is 5 types. So you have got the 5 types of IG. 5 types. IG. Because this one I put it out 1, 2, 3, 4, and 5. 1, 2, 3, 4, 5. There are 5 types of heavy chains, and then that constitute and the 5 types of IG type. Okay, and the IG among this, which I mentioned before, we will go on different types. IgG, right? And that is the most abundant one. This is the most abundant immunoglobulin, and that's also present in the extravascular fluids. See, if you have a blood capillary which is running through, so this one, this IgG type, which can easily migrate and it will go everywhere. And what is the function of IgG? We'll see that one, how this antibody is been recognized by the pathogen, and that they help killing the organisms. Okay, we'll see that one in a minute. It neutralizes the toxins, IgG, and then combines with the microbe by fixing complement via C1 pathway, that's a complement 1 pathway, facilitating the phagocytosis through the what region here, the constant region. FCR, the region we call it the R, FC region. FCR is C for constant and F for the fragment and R for region. FCR responsible for the activation of the phagocytosis, and also in the C3b receptors. And the C3b receptors are present in the phagocytes. So it will activate that C3b receptors or the complement, and this complement binding, and this is also activating to kill the microbes. And then this IgG type has also been crossed with the placenta in the late pregnancy. IgG type can also pass through, and they give the protection to the young one. And it crosses the intestine in the neonate. So when mother fed the children with the breast milk, mother's milk, they contain excess amount of the IgG type, and that can also getting through the mouth and getting intestine, the neonate can get it. So in that way, they call it as if for the nursing person can also get more on the child, the baby get more of the immunity. So that's one of the reason of the Ig type, which is going over there. Now we will see in your textbook, how that's on the figure 315. If you see that, I will check into your explain this picture. This IgG type maternal, okay, this is the placenta. How this is, there's the receptors, this is the FcR receptors, the reaction, and the receptors will bind with the maternal IgG, this is the maternal circulation, that passes through the placenta, and then is getting into the fetal circulation release. So the fetus, they receive this immunoglobulin. The fetus is not synthesizing the IgG type. It is coming from the mother and getting into that. The same thing is here, neonatal acquisition of the milk. From the milk, if they drink milk after coming out, after giving birth, the young one, and that infant also receive this milk IgG. And then here, the maternal IgG is coming from the serum. And then as I mentioned before, this IgG is a good molecule which can extravascular. It can come out of the blood vessels and getting into the biological secretion of the fluids. And that's one of the things, the milk, and through the milk coming to and release the milk into the basal surfaces of the infant. Okay, and this is another one, the acidic response of intestinal self of adult, where the antigen, how this antigen is being transported through this antibody reaction. So we want to know exactly what is the mechanism which is being present in this for IgG transport across the intestine, across the placenta. So that's one other picture. And the next one is the FcR, that otherwise we call it as the constant region, okay, fragment constant region. They have the receptors are specialized for different functions such as phagocytosis. So the receptors, if we activated the Fc region, that activate the phagocytosis. So as I mentioned before, the receptors are present in the phagocytes, right? Then antibody-dependent cellular cytotoxicity and placental transport and beta lymphocyte regulation. The beta lymphocytes regulation, they also have the Fc region of the receptors of this particular antibody, because the B cell lymphocyte is responsible for the activating the antibody, okay? The receptors, just now I mentioned before, the placental transport of the FcR. IgA exists in mainly as a monomer in plasma, basic 4 polypeptide unit. Also it may present as a dimer in seromucous secretions. Exhibits defense of external body surface. So external body surface we have mostly of IgA. In other words, all your sebaceous glands and other sweats and everything, they also have an antibody, IgA type of antibody, which can also prevent the pathogen getting inside. IgM, pentameric molecule, minor fraction is an hexameric. This is a type of molecule, how many molecules constitute form IgM. And they also present intravascular, produce early in the immune response, and bacterial agglutination, and through high valency, it can bind with many IgM molecule combined to the bacteria, and thereby it activate. And then mediator of complement dependent cytolysis may be activating, killing the molecule of bacteria, or killing the bacteria using this molecule. and they have the powerful first-line defense against bacteremia. What is bacteremia means? If you have anemia, you know that, right? And you loss of more of iron, you become anemic. Like the bacteremia means you have more of bacteria present in the blood circulation, in the blood. That's called bacteremia. Okay. And now we got IgD type, and they largely present in lymphocytes. The Ig type present only in the lymphocytes. They functions together with the IgM molecule as the antigen receptors and naive B cells. As I mentioned, this antibody can also bind with certain cells. Do you remember that T cell receptors, like I mentioned before? So certain type of antigen, they can also bind, the certain type of antibody can also bind with a specific cells, B cells and T cell. And the IgD molecule is an antibody, IgD or immunoglobulin D, can also bind with the naive B cells, and thereby can activate the synthesis of more of immunoglobulin. IgE is present in a firmly in the most of the cells, and they contact with the antigen, it will activate, and the local recruitment of antimicrobial agents through degranulation, release inflammatory mediators. IgE responds for certain parasitic infections, responsible symptoms of atopic allergy. So IgE is the one which is activate the allergic reaction. If you have a skin, it's a mosquito bite or any other sting, any other itching, if you're getting onto the skin, and those are the allergic reaction which will activate mostly because of the IgE responsible for that one too. And this diversity of function is possible through the subdivision of classes into subclasses and based on structural differences in heavy chains. So all these functions with the IgG type, which I mentioned, IgG, IgM, IgD, IgA, so all these, it's a variation on the, why it is a variation of different type of IgG? Because of the heavy chain molecules. So now these heavy chain molecules are based on structural difference on the heavy chain molecules. And all these different varieties of Ig type, which is present in the normal individual. Okay. So it may vary if there is a disease, you have more of IgM or IgD or IgA. For example, IgE is responsible if you more of IgE synthesis is gone up whenever you get allergic reaction. Certain type of parasitic infection, you may have IgD. So we can determine if you have a profile of immunoglobulin in your normal profile of immunoglobulin will give a diagnostic value for a particular type of infection or disease. So that's the other things you have to understand. Okay, this is a part of the PowerPoint, and I want to recollect and some of these.
The basic immunoglobulin structure consists of 4 polypeptide chains, 2 identical light chains and 2 identical heavy chains, joined together by interchain disulfide bonds. Amino acid analysis shows that the N-terminal portion of the heavy and light chains has a variable composition when different immunoglobulins are compared. We talk of this portion as being the variable region, and we have a variable region of the light chain, designated VL, and the variable region of the heavy chain being designated VH. Amino acid analysis shows that the remainder of the peptide chains, both heavy and light, are relatively constant in composition. Again, we designate the constant region of the heavy chain as CH, and that portion of the light chain, which is relatively fixed in composition, we refer to as the CL region. Certain sequences in the variable regions in different antibodies show quite remarkable diversity, and these hypervariable sequences have been localized to 3 segments on the light chain and 3 on the heavy chain. We refer to these segments as complementarity determining regions, or CDRs, because they're the portions of the antibody molecule which make contact with and are complementary to the shape of the antigen against which they're directed. The immunoglobulin G molecule consists of 1 such 4-peptide unit. The first type of variation we'll look at is termed isotypic. All isotypic variants are present in the serum of a normal individual. The major immunoglobulin classes are a good example of isotypic variants. These are located in the CH region, that is, the constant region of the heavy chain, and the different classes are IgG, M, A, E, and D. These different structures give rise to separate and distinct biological functions. Within the overall structure of a given immunoglobulin class, say IgG, there can be further minor variations in structure which give rise to subclasses. In the case of IgG, these are IgG 1, 2, 3, and 4, and again, this leads to some differences in biological function. There are two isotypic variants in the constant region of the human light chain, and these are labeled kappa and lambda. The lambda isotypic variant of human light chain can be subdivided into two different subgroups, OZ plus and OZ minus, which represent a similar relationship as the subclasses of immunoglobulins do to the classes. Lastly, within the variable regions of the heavy and light chain, there are isotypic variants called subgroups, and in essence, these are different framework structures which carry the different hypervariable regions constituting the antigen combining site. The next type of variant we wish to discuss are termed allotypic variants. These are alternative forms, or allelic forms if you like, of an immunoglobulin which are genetically encoded and are not present in all individuals, just as different alleles are not necessarily present in different individuals. These genes give rise to what are termed allotypic immunoglobulin variants, and these occur mostly in the constant regions of the heavy and of the light chain. One example of heavy chain allelic forms is provided by the human GM groups. These differ from each other in minor changes in amino acid sequence within the FC region of IgG. Lastly, we come to the idiotypic variants, which are individually specific for each immunoglobulin molecule. In essence, these are shapes characteristic of the hypervariable region which form the antigen combining site. These shapes are recognized, just as an antigen shape is recognized, by an antibody or an antiserum raised against that shape. We talk of the original immunoglobulin as an idiotype and the antiserum raised against it as an anti-idiotype. In fact, they mutually define each other. You cannot have an idiotype without its defining anti-idiotype serum, in the same way that it's impossible to talk of an antigen without referring to its antibody or appropriate T-cell. Thus, the idiotypes are located on one or more of the hypervariable regions, forming the antigen combining site. To summarize, isotypic variants are seen in the constant region of both heavy and light chain, and also as framework structures within the variable regions. Allotypic variants are present largely within the constant regions of the immunoglobulin chains, while the idiotypes are located essentially in the hypervariable regions. IgG is the most abundant immunoglobulin in the internal body fluids and is present in high concentrations in the extravascular compartment, where it combats microorganisms and their toxins. Although present as a monomer in blood, where its function is not entirely clear, IgA is found mostly as the major immunoglobulin in the seromucous secretions, in, for instance, the lung, saliva, and intestine, where its job is to defend the external body surfaces. IgM is essentially confined to the vascular compartment, the blood compartment, and due to its multivalency is very effective at agglutinating bacteria. It's produced early in the immune response, where it's an effective first-line response against the presence of bacteria in the blood. There's very little, if any, IgD circulating in the body fluids. Most, if not all, IgD is present on the lymphocyte surface as an antigen receptor. IgE is a second-line defense for the external body surfaces, where it acts as an initiator of acute inflammatory reactions through its binding to mast cells, thereby recruiting antimicrobial agents to the site of infection. IgE levels are raised prolifically in parasitic infections. Also, it is the particular immunoglobulin which bears responsibility for mediating symptoms of atopic allergy. IgG, and even more so IgM, are the most effective activators of the classical complement pathway, combining with the first complement component C1. IgA, less effectively, is the only one of the immunoglobulin classes which can activate the alternative pathway, which depends upon stabilization of the C3 convertase C3b-BB, which usually occurs independently of antibody. Only IgG crosses the placenta to the fetus, thereby providing passive immunity in the neonatal period. IgE binds with great tenacity and high affinity to the receptors on the surface of mast cells and basophils, which recognize the Fc portion of the immunoglobulin. One of the subclasses of IgG, IgG4, binds with some affinity to mast cells, but is a far weaker interaction than that observed with IgE. On the other hand, the IgG-Fc region has a strong affinity for the specific receptors on the surface of macrophages and polymorphs. This binding is especially strong when the IgG is in a multimeric form, as in an immune complex, when, in addition, it cross-links the surface receptors and initiates phagocytosis. IgA binds to the macrophages in the external body fluids. Recently, it's been shown that IgE combines with specific receptors on macrophages. Here we are looking at the two heavy chains and have picked out the N-terminal VH variable region domain, the first constant region domain of IgG, C-gamma-1, the second, C-gamma-2, and the third, C-gamma-3. Note the way in which the two C-gamma-3 domains are binding together. They do this non-covalently through conserved residues on both domains and bind firmly. C-gamma-2 residues are held apart by masking of the hydrophobic regions by carbohydrate. The resulting independence of the two C-gamma-2 domains allows the peptide chains linking them to the C-gamma-1 domains to be highly flexible. This provides the flexible hinge region, which enables the combining sites in IgG readily to adapt to spatial variations in the location of the antigenic epitopes, which react with the combining sites. We now put in the two light chains, showing the variable VL domain and the constant CL domains. The CDR complementarity determining regions in the VH and VL domains provide the antigen binding site. Sites on the C-gamma-2 and C-gamma-3 domains bind to the Fc receptors on macrophages and polymorphs, and natural killer cells, NK cells. They also bind to the placental syncytiotrophoblast to provide the transportation of IgG across the placenta and into the fetus. The C-gamma-2 domain provides the site for fixation of complement component C1q, which initiates the classical complement pathway and also controls the catabolic rate which determines the half-life. Note the inter-domain pairing between VL and VH on the one hand and CL and C-gamma-1 on the other, much the same as the non-covalent bonding between the two C-gamma-3 domains mentioned earlier. When the IgG molecule is split by papain in the hinge region, one can see the structure of the resulting two Fab fragments, each of which is univalent for antigen binding, and the Fc fragment with the C-gamma-2 and C-gamma-3 domains, which provide the biological characteristics of the immunoglobulin class related to the particular heavy chain structures.
Just getting onto the home page. Okay. It's not coming up now. Look out. It's not coming up, right? Anyway, just I want to get, okay, this one, So I want to get through the summary of what we have learned on this antibody class about it. Do you have any questions so far? The first part, which I showed you about the mechanisms and the basic concepts, and second part, which is of the PowerPoints, which combined together, and then the third part, which is the review from your site. You can also listen to those talking PowerPoints from the publisher site at your leisure time, and then to try to understand the mechanisms completely. And for your exam next week, I want to review that thing. If you go on to that site, how many of you visited this site? Right? Okay, good. So if you go on to chapter 1 to chapter 3, see chapter 1, innate immunity, specific acquired immunity, chapter 2, chapter 3 is antibody, okay. The chapter 3, if you go down, if you see the MCQs, could you see that one? I'm sorry. It's mine. Okay. If you see this one, the MCQ on this, multiple choice question, click on that and you will find the basic Ig unit is composed of, just now we learned how many. Basic immunoglobulin unit is composed of 2 identical heavy chain, 2 identical light chains, 2 identical heavy and 2 different light chains, 2 different heavy chains and 2 identical light chains, 2 different heavy and 2 different light chains, non-covalently bound polypeptide chains. Which one is the correct answer? Okay, that's good. Submit the answer. The information you have entered to be sent on over encrypted. Continue. That's correct one. Okay. So then you can go for the next question. FAB fragment is produced by pepsin treatment. Separation heavy chain light chain. Binding antigen lacks light chains. Has no interaction interchain disulfide bond. Which one? It's produced by, let me see. See? That's correct. It's incorrect. Pepsin is incorrect answer. Binds with antigen? That's correct. Good. So you have to go through those questions for every chapter. And the complementarity determining regions are restricted to light chains, are in the constant part of Ig molecule, or bind to Fc receptors, are concerned in antigen recognition, occurred at C-terminal end of Ig peptide chain. Which one? The complementary determining regions are concerned in antigen recognition. Yes, that is correct. 3 CDRs, just now we saw that one, okay. So please go ahead and get the answers, correct answers, and study, review from your class notes as well as from this, and innate immunity, specific acquired immunity antibody in this MCQ, number 1. Number 2, you have done your homework for the chapter 1, chapter 2, right? From that, from the Zoom book you have done. The same thing with the chapter 3, you have on the page 53, okay, this book, okay, let's go back, okay, and page 52 and 53, you have review questions, and these answers are there. So I would like to go through this question, okay, for your exam to study. Some of the questions is coming from the MCQs, and some of the questions coming from the short answer questions from the Zoom book. Okay, chapter 1, 2, 3, and I have given you the homework 1 and homework 2 you have done, and then for the chapter 3, which I have not given anything, don't worry about it. You study these questions for the exam. That is your homework, because I don't want to give you any homework now in this one. If you have it, if you write it on your homework notebook, question answers, that will be fine. I will get back the questions and answers to your homework book at the end of your exam next week. okay, so that I will evaluate your homework as well as together and then give the grade, how far you are doing that. It's fine. For the Victoria and Sugarland, Victoria and Cinco Ranch, could you submit those papers of your homework and you submit into your, I think there is a folder there, you can submit there, I can collect it from the office, or the homework 1, 2, 3. The same thing with Victoria, homework 1, 2, 3, please submit there in their box, which I think it is in Victoria, so I can collect it from them. Okay?
Dr. Soma,
Yes.
this is Ellen at Cinco Range.
Right.
You originally told us to do the homework in a notebook.
Yes, that's on a notebook. You have done that one, you can submit the notebook. Okay?
Are we
I
submitting it today or after the exam?
After the exam. So you will have an exam as well, okay? You have the exam evaluation, your homework evaluation together will be there. Okay, you can study, because I want you to study those question answers as well when you write it. Okay? Any questions? That's for the review of the question as well as the homework. Any questions? Or do you follow anything? Are getting confused? How many of you got confused? No confusion?
Where I leave my homework.
You want to leave your homework next week after that exam. There is a folder in the help desk onto my name in it. Otherwise, I'll ask them to collect it, and then they will send it to me. Perhaps you can check with Karen Pierce there, in the biology technician. You can check with her and then ask her to send it to me so that I will evaluate here. Okay? Either in the paper or in the book, notebook. And then I will send those things out the following week so that you will receive those notebook back after evaluations. You can read and you can understand and you can write on your answers on the book. On your own words, that's fine, including the match the following, everything. But some of the answers, if you write it, just copy down the book. Doesn't matter, you can also do it. I want you to write it, not type it. You can't type, you can type it, that's okay. Just I want to do it anyway. I want to write it on the notebook. Any other questions? And this exam is going to be onto the computer. You have to type the answers, okay? And it is going to be a closed book exam, not an open book test. Okay? If computer fails, then I will ask you to write it on a Scantron or on the paper copy I will provide to you. Either way. At the same time, the exam is going to be there, and once the exam is over, then if you have time, then we can go ahead to have the next chapter we will discuss in the class. The same classroom. I will bring the computer to you. Okay? I'll bring the computer to you.
You also know the papers, the research papers.
Oh yes, she reminded me. I think in your syllabus you find there's a paper pending. There are three titles which I given to you. So I'll reduce to one because we are doing this homework. So out of 4 or 5 topics, you can select one topic and write your research paper.
Thank you. Yes.
Okay? That's the part is the online teaching. I gave that one because they have to work on the computer. I did, but if you want a face-to-face class, I want to reduce that assignment to one because we are going to deal more on the discussion part probably next week. Okay? If any other questions?